Alright, so first off, this presentation is for the general audience and it's not in detail, just the basic stuff. And we're going to look at what gene therapy is and specifically how do viruses contribute towards gene therapy. And then at the end, we're going to look at different therapies in practice, and then we are going to look at the limitations and future work that is being done in gene therapy. So let's start. Gene therapy is basically introducing a gene to either treat a disease that someone is born with or to treat a disease that someone acquires. For example, it could be a case of hereditary anemia, or it could be a case of acquired infection. For example, I'm not so sure about whether there is a gene therapy for HIV, but let's say if I have acquired an infection like HIV, then I might use a way to basically disrupt the function of a particular gene that might be involved in the spread of the disease. So when we talk about gene therapy, we always go back to the central dogma of biology, which we have always studied in almost every subject of biology, that DNA leads to RNA, which leads to protein. What's important about this is that the end product of a gene is basically your protein, and the disruption in that function of the protein depends on the type of disease that occurs. So basically we can say that when we have a disease, it could either be due to a protein that is non-functional, or it could be due to a protein that is functioning in the wrong way, or it could be that no protein is being produced, or proteins could be produced more than they're required. So in all sorts of cases like these, we need to look at gene therapy for a cure. So the cure is basically to introduce a gene that is going to rectify the problematic protein. And the reason is that because a protein could be immunogenic. The term immunogenic applies initiating an immune response. So certain proteins could initiate an immune response. It could lead from mild to severe anaphylaxis, which is the medical term for inflammation. In order to solve this problem, we look at the type of gene that is producing that protein so that we can insert in the human body. The first approved clinical gene therapy was done in the 90s to treat severe combined immunodeficiency, abbreviated as SCID. However, this therapy was not successful. So now that we have a general idea of what gene therapy is, let's talk about the two types of gene therapy.